embedded in origins is the fact that evolution proceeds, as we speak. And you would say, well, how is that possible? So this is kind of what I'm going to be presenting to you today. And it's related to the COVID-19 epidemic, the epidemic calendar. And I'm going to talk about the molecular impact of the COVID-19 epidemic calendar, and it's going to be about evolutionary genomics, but it's going to be also about molecular structure. So those are the two aspects of my presentation today. Let me see if I can advance, because I can seem not to be able to advance my slides. Okay, oh, there we go, yes. Okay, so what is the epidemic calendar? Well, you may know the epidemic calendar with other names, scientific name is seasonal forcing, but in the public domain, it's about seasonal behavior. And we know this very well because we are exposed to the flu, and the flu virus, evolves very quickly, and therefore we have to give us flu shots every year, in order to counteract the disease. So this is about virus evolution, because the virus obviously evolves, and that's the reason why we have to, act, with winter virus in the way we do. Now, I say virus evolution, but saying virus evolution is problematic, because there's been a big controversy, and the controversy is whether viruses are alive or not. And if they're not alive, which is the standard kind of view of viruses, these are kind of rogue particles that appear and make our life miserable, then if they're not alive, how could they evolve? So in my lab, we've been pushing this concept of viruses being part of the makeup of life. And because we think that they are alive, we've placed them in the tree of life. And so here you have a tree, and in this tree, which is a description of relationships of things, it's a diagram. And the diagram shows a distance between the individual endpoints, which are the leaves of the tree. And those are viruses, archaea, eukarya, bacteria. Those are the organisms that make life in this planet. And the distance between them indicates whether they're closely or distantly related to each other. But because this is a rooted tree, therefore, we can say viruses are primordial. And we've been pushing forth this idea that viruses are at the origins of life, and that viruses have cellular origins. So this is a process of billions of years of evolution. And now I'm going to jump to a process that basically is going on every day. And this is the COVID-19 epidemic. The COVID-19 virus, called SARS version 2, is a virus that has been changing notably, and we can look at planetary snapshots of this virus that is covering our planet, and if we can place that tree that describes the relationships of the different viruses in a circular diagram or circular tree like the one you have there, in the middle of that tree, you have the origin of the epidemic, the virus that was sequenced in Wuhan when the epidemic started in 2019. And as the virus has been progressing, each concentric circle is an addition of time. And so you have there a sampling of individual viruses at different times of the pandemic since 2020 till February 2022, where I just decided to take this kind of planetary snapshot of the viral population. And each of those dots represents a genome, and the tree is built from the differences in terms of mutations between the genomes. And you can see in this tree something interesting. You see there at that time, this was the start of the Omicron virus variant that is currently the one that we have in the planet. And you can see how in just few months, it just basically covered on, on that edge, the majority of the genomes that were sampled at that time. And this is a newer one, November 2023. This is the new tree. And as you can see, now we abandoned the initial variants of the Omicron, and now we have new ones. We have the Arcturus, as have been called in the press, which is one that's taken over this variant, the planet. But now we have also the Pirola that you have there. You can see, I don't know if you can see my marker, but there you have the Pirola alternative. This is a variant of concern that is appearing and there has new mutations, and perhaps it overtakes the planet. We don't know. That we had another one called Centaurus. Now, one thing that is interesting about this diagram is that it brings us back to Darwin. And if we think that this would be a tree of life, it's a tree of the COVID-19 virus. But if that would be life in our planet, then Darwin's intuition that the great tree of life fills with its dead and broken branches the crust in the middle of that diagram and covers the surface with its ever-branching and beautiful ramifications. As you can see, all this diversity that is unfolding in the outskirts as the virus evolves, then we are basically confirming in real time, because this is real-time biology, one of the great statements of Charles Darwin's On the Origin of Species by Means of Natural Selection book. So I was bring this because it's an interesting observation, just to see how all these trees that describe diversity of the virus unfold and how that is giving us information about evolution. So what are my objectives today? Well, I want to explore questions. Genomic change, we know, occurs at planetary level with the virus. Why? How? Where? When? We need to ask that kind of questions. So I'm going to explore with you genomic diversity of the evolving virus. And we are going to explore seasonal behavior and its molecular culprits. And one of my goals, and this is one of my interests for many years, is the exploration between the link between structure and function in biology. And we do that with the proteins of the virus, of course. What is the goal of this type of research? Well, I would want to develop predictive intelligence. What does that mean? Well, use information that we may have, like for instance, we've been mining structure of molecules with a very large supercomputer on campus that's called Blue Waters, and we've been simulating, molecular dynamic simulations in this frame, and we want to use that kind of thing and link it to systems biology approaches. And this kind of information that I'm going to show to you, and then develop predictive intelligence tools that we can then test their accuracy with, and then use all this information to mitigate current and future pandemics. So we want to predict when is it that a mutation will occur, and when is it that these changes will occur in a virus as a pandemic or epidemic appears. So the focus is going to be seasonal behavior. And SARS-CoV-2 is a winter virus. And winter viruses like the flu or the swine flu are viruses that manifest their infectivity during the winter time. So it's a seasonal effect. And you can see the seasonality of the virus with my example there at the top, the swine flu. in the city of Chengdu in China between 2007 and 2016. And there was a pandemic of the H1N1 virus in 2009. And you can see that at that time, we had semi-annual cycles that then turned into annual cycles of the disease, and then after some time back again into semi-annual cycles. Why? Why is this? You know, this is kind of a magical thing that the viruses will come and go in terms of their infectivity. And there are many, ideas about this, but we know that the virus somehow interacts with the physiology of the host on one side, but it does so with the environment of the host and also with the behavior of the host and the virus. Like, for instance, measles usually appears during the school year. Why? Because kids get together and they facilitate the transmission of the measles virus. So a behavior component is important, but there's also an environmental component, and it's difficult to track. And this occurs not only with viruses, but with many other pathogens, microbial pathogens. So when the disease hit, and this was a research that started in my bioinformatics class here at the University of Illinois, we got interested in determining whether COVID-19 was going to be a seasonal virus or not. And there are different ways to uncover seasonality. One is called spatial distribution approach, and that implies, when you have a broad distribution of a virus in the planet, you can actually divide, some epidemiological parameter, like a number of cases per million people, like the one you have there in the diagram, and then use Pearson correlation or even Spearman correlation to determine whether there is a correlation between the place in the world when this is happening and therefore the location and temperature of that place, let's say the latitude and the temperature of a country or a city, and then determine whether there is a correlation, global correlation with epidemiological parameter. And what we did at that time, we did this very early in the pandemic. It was in April 2020. The virus has just arrived the shores of the US, and it was going to wreak havoc in our country. And it was interesting because we found a correlation. It was mild, but the correlation was there. We repeated this in 2021. Data was 2021, and the correlation got stronger. So we had already an indication that COVID-19 was seasonal. We saw, you can see basically the seasonality unfold as the disease was swiping the world. It started in the northern hemisphere in a corridor, a very defined corridor, and then spread nicely all the way to the southern corridor in the other hemisphere. So it was changing as it was developing. It was looking for the right environmental area to unfold. And the work in China from Liu and colleagues has used an alternative to this, which is called the temporal distribution approach. And in this case, what you do is basically use statistical time series analysis and statistical approaches that are used to model your data, and using cases and deaths, for instance, in this case, and see whether you have, as the disease progresses, a synoidal pattern like you have here that is shifted in the different hemispheres of the world, and that will be an indication that we have seasonality. So this work that was unfolded during the first year of the pandemic in our lab and many others, and that revealed that indeed the disease was seasonal. So we had that interesting component there. We confirmed very recently the correlation, and so this is a time series showing how the Pearson correlation, the Spearman correlation, both of them kind of stabilized with time and became strong. So showing that the disease is indeed seasonal. So The other thing that interesting that we found out is that we were looking for other correlations that would be of importance. And one of them that was interesting was correlate the Yale's EPI, it's called Environmental Protection Index. What is this? This is an index that that is used by the United Nations and many countries to evaluate how well we are doing with planetary change. And it has two general descriptors: an ecosystem vitality, these are objectives, ecosystem vitality in the center here of the pie chart, and environmental health. And then there's a number of descriptors in the periphery, for instance, CO2, ammonia, nitrogen, wastewater, stock status, for instance, grasslands, PM25 exposure. So these are contaminations, for instance, household solid fuels. So all these are components of this index that measure how well a country is doing in terms of combating planetary change. But what we saw is that there was strong correlations of epidemiological parameters, total deaths and total cases, with all these indices. And so seasonal behavior was also linked to global change. So it was affecting our planet in some way. The virus was affecting our planet. So let me see. Okay. So let's go now to the genome. The genome of the virus is 30,000 nucleotides long. It encodes 29 proteins. These 29 proteins are divided into three groups: the non-structural proteins in the left in blue, and the structural proteins, we know some of them very well, and accessory proteins. The non-structural proteins are important because they make what is called a replicase. This is a complex that once the virus enters into the cell is responsible for making copies of the virus. And it requires all this number of proteins for this to occur. Now, at the center of it is NSP12, there at the middle, roughly. That protein is responsible for making copies of the RNA of the virus. In the right, you have the structural proteins. And the structural proteins, what they do, they are responsible for making the capsid of the virus, and for holding the RNA makeup in the middle of it. And so you have the nucleocapsid, the spike proteins, which is responsible for that crown appearance of the virus, but also the envelope and membrane proteins. So this gives you an idea of the of what we are dealing, basically. So in my talk, I'll be talking about 4 genomic exploration of viral diversities, and these 4 genomic exploration occurred throughout the pandemic. The first one was at the beginning, in which I did with my students. We explored around 15,000 genomes in May 2020. Then later on that year, we explored almost 150,000. And then more advanced in 2022, 7 million genomes. And in July 2022, 12 million genomes. And as you can see, as that exploration was unfolding, we were exploring different things. The first wave of the pandemic, the alpha wave, the delta wave, and the Omicron wave. And you can see in the diagram in the bottom, how is it that the different variants of concern have been overtaking the planet, as they were advancing. These are the 4 major, foci of research. The beginning, we were interested in protein flexibility and intrinsic disorder. Then we wanted to know what was the culprit of seasonal behavior. Then we were interested in haplotypes. and I'll define them in a minute. And we started by studying them in Australia. And then the last exploration, which we still have papers to publish on this, we went into the structure and studied the structural changes of those mutations and what they were doing. So, so let me just first explore some definitions, which I think it's important so you can understand some terms that I'll be using. So what is a variant? You know, a variant is a virus that holds either one mutation or a unique constellation of mutations. That's the terminology, constellation of mutation, a group of changes in their genomic makeup relative to something. Everything is relative, you know, in biology often it's relative. And here it's relative to the reference strain. The reference strain is that first Wuhan strain that was sequenced when the disease started in the city of Wuhan in China. Now, this variant is different. That's a virus, but we also talk about amino acid variants. And what are they? Well, these are mutations that cause alternative amino acid substitutions in a protein. So if you have a sequence of amino acids, because proteins are made of sequences of amino acids like the ones you have there at the right, and you compare those sequences to the sequences of other viruses, and you see a difference there, you know, then you will have what's called a substitution of one amino acid by another, like the one you have there, an isoleucine by a threonine in sequence variant 7. So sequence variant 7 has it's an amino acid variant because it has that substitution. And you can see also an insertion there in the middle in sequence variant 2, where you have 2 amino acids added in a particular site in that genome. Now, we used a specific nomenclature, you know, that is established that, like for instance, if we know the location of that mutation, let's say it's in the spike protein in location 614, then we say that at that location there was a substitution of an aspartic acid to a glycine, and we call that a D614, that's the location, to a G. So that means the D was changed to a G, the aspartic acid was changed to a glycine. And that's kind of the nomenclature that we have for mutants, and we've been exploring using that nomenclature to define the thousands of mutations that we've been exploring. So now let me define some other important things. What is a mutation of concern? A mutation of concern is a mutation associated with statistically significant and experimentally verified increase in either virus transmissibility, disease severity, immune and vaccine escape, etc., etc., etc. I mean, these are all things that are of clinical or epidemiological importance for us. Now, for instance, if a mutation makes the virus evade a vaccine, that's a terrible thing to occur, and we want to know about that. So that's a mutation of concern if we identify that. Now, what is a variant of concern? Well, variant of concerns, they're called VOCs, they are variants that exhibit constellation of these mutations of concern. So whenever we have many of those appearing in a variant, then we get really concerned. And we know about these ones because they have this Greek letter nomenclature, alpha, beta, delta, and so on. And some of them have been terrible for us in terms of deaths occurring in our planet. And then we have this other interesting concept, the concept of haplotypes. For you in genetics, you know what this is. But what is an haplotype? It's sets of mutations that are inherited together. But a virus is not an organism. So viral haplotypes are mutations that appear often linked to each other in viral variants. So whenever 2 things always appear, you know, at the same time, that becomes an haplotype. And we are very interested in them because they make the mutant constellations. And we need to understand why the mutant constellations appear in evolution. So from a bioinformatic point of view, and remember I'm a bioinformatician, what we do is to build this what's called multiple sequence analysis. So we align different sequences to each other, And this is a bioinformatic exercise of finding what is called amino acid correspondences. So we want to find where is the isoleucine in that alignment, and then make a statement about how that is correlated in other different sequences. And this is how our analysis is going to proceed with this kind of approach. So in our first exploration, we were interested in pathways of molecular change unfolding during the first wave of the pandemic. And remember, we had about 15,000 sequences to analyze. Those are the sequences that we had at that time. There are no, just the laboratories in the world were starting to sequence the virus and getting set up in all countries. So we decided to use two measures: a measure of entropy, which is a measure of information that measures diversity, and an entropic delta, as we call it, which measures evolutionary constraints. And we looked and screened all mutations that appeared. There were tens of thousands of them, and we were exploring whether, according to some criterion, which were the most important. And we found that there were only 27 amino acid sites that features these entropies that were above 0.1 bits. And those were the ones that we were very much interested, because those were the ones that were unfolding in the planet as some target, for some reason, and wanted to know why. And these changes affected 13 out of the 29 proteins, meaning that only just a fraction of the proteins of the virus were being modified. We found a total of 19 sites that engage in reversals, suggesting tendencies towards mutation fixation. But then there were 8 sites that showed tendencies of entropy expansions that suggested that the virus was diversifying and it was drifting, in terms of the makeup. So we found 2 completely different opposing behaviors. But one thing that was very interesting, and remember this was done at the very beginning of the pandemic, is that we found 4 mutations that spread throughout the regions of the world, and they form 2 haplotypes, meaning that the mutations, whenever there was one mutation, the other mutation will appear at the same time. And you may have heard about this, that the spike protein, the mutation in place 614 of the spike that had changed to a glycine, that mutation appeared together with a mutation in the RNA polymerase of the virus. That's in the right-hand side in red on the top. And those 2 were always linked, where whenever that 614 mutation will have the 323 mutation in the polymerase appearing, plus 2 other mutations that did not change protein makeup. They always appear together. The other one that appeared always together was in the nucleocapsid protein, the protein that wraps the RNA of the virus. And this appeared in a region that was of very much interest to us, and again, it spread throughout the world, and those 2 haplotypes are still with us today. And the reason why we were so interested and excited about this result is that because those 2 mutations were showing there were early pathways associated with flexibility and intrinsic disorder in proteins. The 614 mutations in the spike in the left, and there you have the spike protein, and beside it the RNA polymerase, were in locations that were important for flexibility. That mutation is known to enhance flexibility in the spike proteins, and the mutation in the RNA polymerase is an interaction junction between the interface, in the interface domain that spans 2 important domains of the RNA polymerase, one of them called the thumb. And this was an interesting indication, but when we looked at the N protein, we saw that again the mutations were located in areas where the protein had a lot of intrinsic disorder unfolding in their makeup. Intrinsic disorder means that that region of the protein does not have typical protein structure. is highly disordered area of the protein. So we were very curious about this because intrinsic disorder has been linked to, in epidemiology, to disease severity. And so we got so excited that we said, well, let's explore how coronavirus in general fare in terms of disorder with all viruses that are known. So we took all genome sequences of viruses that are known and calculated mean disorder for their genomes, well, the proteins that the genomes encode. And what we found is that the coronavirus and even the nidovirales, which is the viruses that hold the coronaviruses, all of them had a considerable, were really rigid. They didn't have too much disorder. And the disorder was really localized in particular regions. Like, for instance, this HVR region of the NSP3 was, and on the bottom there was the only one that had a disorder area. The score was really high. So rigidity, and there have been a lot of work on this, allows viruses to withstand harsh environmental conditions. So if you have a lot of intrinsic disorder in your proteins, then your proteins are very flexible, they're not rigid, and they're interesting. They provide possibilities that rigid proteins don't have. But then in terms of a virus, they will not protect the virus against harsh environmental conditions, like temperature and humidity. So this is interesting, isn't it? Because we are seeing that mutations are occurring in places that do not help the rigidity. They're trying to foster flexibility. So it goes against the idea that the virus is trying to increase the virulence. This is something interesting, an interesting observation. So some conclusions, very quickly, I'm going to go through of that first study. SARS-CoV-2 is a winter virus. It exhibits disease seasonal behavior. There are evolutionary forces changing the genomes of the virus. There's a balance between fixation and diversification and drift. The early drivers of mutational change were related to proteins, flexibility, and intrinsic disorder. And viral survival was somehow linked to virus rigidity, which facilitates persistence in harsh environments. So in the second exploration, we were interested, and we were still in 2020, and we were interested in exploring how did mutations manifest in the genomes. And so we had two expectations, that there would be steady accumulations like the ones of the 614 mutation of the spike, or there would be mutations that would appear in burst and then disappear. We didn't know really what we were going to find, but we were expecting that both types would appear as we were exploring this larger pool of genomes. And we were interested in focusing now on the spike, because the spike was the focus at that time of vaccine development, and we didn't have vaccines. And we thought, well, let's explore if it's the spike is antigenic, where would mutations appear, in the spike. And the spike, as you see in the bottom, has two regions. It has what is called the S1 subunit in the left and the S2 subunit in the right. And the S1 subunit is the globular part on the top there on the three-dimensional structure. It has the RBD, as it's called, is the binding domain, and the N-terminal domain, which both of them are important for binding and are both important antigenically. And then you have the other regions at the bottom. Those are the regions that are embedded. Remember, this is a protein that is embedded in the membrane. So it's all helical regions of the protein that are known to interact with the phospholipids of the membrane. So we wanted to know whether mutations appeared and how did they appear. And what we found is that the viral population evolved via mutational burst. So you can see how the disease progresses in this diagram. It's a heat diagram. So, and you can see how the darker hues implies that the virus is spread throughout the world. And you can see on the top the D614G mutation spread and continue to be prevalent throughout the world. But then the other mutations that would appear, they would appear and go, come and go. And some of them will be revisited, but but they will then disappear. The other thing that we found in our analysis is there were mutant constellations already being formed. Even though we had only that 614 mutation in the spike, the only one that was preserved, there were just groups of mutations that appeared together, and their prevalence was increasing. At that time, we found that on average there were 2 mutations per virus variant, but there were some viruses that had up to 9 mutations. And this is the beginning of the pandemic, isn't it? So, but the one thing that was interesting is that we found that in all these combinations, the majority of combinations was between that 614 mutation of the spike there at the center of that graph, and mutations in the NTD region, the N-terminal region of that globular component of the spike. And so that got us thinking that perhaps the NTD had something to do with the infectivity of the virus. And because we were interested in seasonal behavior, so we explored where were the mutational bursts occurring in the world and whether there was a hemispheric pattern behind it. And indeed, there was. The NTD mutations, the incidence of them followed hemisphere-related seasonal patterns. And so when they were high in the northern hemisphere, they were low in the southern hemisphere and vice versa. So this gave us the indication that perhaps we had a culprit. The N-terminal domain was a genetic culprit of the disease, perhaps a likely culprit of seasonal behavior. So we got interested in what was the NTD doing. And one thing that you do if you're a structural bioinformatician is to produce this kind of diagram. What is this diagram? It's a neighborhood analysis of a particular protein. So there you have on the right of the diagram, you have the spike of the SARS-CoV-2 virus. And the C-score describes how much of a protein aligns in 3 dimensions. Like you have there at the right an aligned region of a protein in yellow and the spike protein. When you align them, if the alignment is excellent, then the RMSD value on the left-hand side, the y-axis, is almost 0 because the alignment is perfect. The larger that number is, the worse is the alignment, and the more distant phylogenetically and structurally the protein is. So as you can see in blue, all of these are spike proteins from different viruses. That is not the SARS-CoV-2. There are many viruses that have spikes. So and as you can see, all of them are kind of related to each other. But then all of a sudden there's a discontinuity, and then you have that region there that is a small region that is labeled in red, and that red region is another different protein called galectin. It's a protein that humans have and that many different organisms have, and apparently that protein aligns perfectly with the NTD domain, which is this domain that has seasonal behavior. So what are galectins? They're a family of evolutionary conserved effector proteins that regulate many processes, and including splicing, cellular interactions, pathogen recognition, inflammatory responses. But we were kind of incredibly surprised when we learned that galectins are important in temperature-dependent coral bleaching, which is a problem of planetary change. We're losing our corals because the innate disruption of a symbiosis between the coral and these microbes called dinoflagellates that populate the seas. And this was temperature related. So there was a culprit that was temperature linked, and the NTD region that we are claiming is seasonal behavior related, that is also temperature related, because seasonal behavior is temperature linked. So our big conclusions were that virus evolved via mutational bursts, that we were seeing the rise of mutant constellations occurring very early during the pandemic, and they were linked to the NTD and the interface regions of the spike, and there were hemisphere-related seasonal patterns claiming that NTD link wax and waning of mutational bursts, and also that the galectin-like culprit of seasonal behavior is this perhaps NTD region that senses the environment and regulates infection. So we were excited with all this, and this poised us to explore more, what was happening later in that year. At the end of 2020, we start having what is called the variants of concern that I mentioned to you, with the rise of alpha first, and then many others. And then at the beginning of 2021, if you recall, that was the vaccine introduction. That's where vaccines started to play, and we proposed three phases in the pandemic: a flexibility rigidity phase, an environmental sensing phase where we were seeing all these NTD link mutations, and then an immune escape phase. And why? Because we were seeing mutations that were affecting the RBD. Remember, the receptor binding domain, the other domain of the spike protein. So we started seeing larger constellations. And there you have alpha, beta, gamma, and delta constellations on the top. And this is the time when VOC delta, variant of concern delta, overtook the planet, taken with this variant innumerable deaths, because VOC delta was much more infective and noxious than the rest. So there was an assumption there implicit in this big constellations that this constellation was stable haplotypes. And we were interested in haplotypes because we were interested in finding whether there were haplotypes linked to seasonal behavior. And so this, in our third genomic exploration of almost 7 million genome sequences, we focus on Australia. And why Australia? Well, this is the map of Australia, and it's a spike maps of where the population in Australia is located. And it turns out that the different states of Australia and territories of Australia have cities located in a beautiful transit of latitude. So for instance, Northern Territory, the majority of population is well in the north of the Australian continent. And of course, 8 is Tasmania, the island at the bottom, and the population there is quite segregated from the population of 4, which is West South Wales and Melbourne and so on. So we had a beautiful transit to explore. On top of this, Australia was wonderful because it was a country that had incredible tough regulations about exchange of people between the territories and states. So it was one of the harshest policies in the planet that were unstated in order to control the disease. So we found that there were haplotypes emerging in Australia. And this was at a time where Omicron was starting. It was just at the time when we collected the samples, 2 months after the Omicron was first detected in South Africa and Botswana. And so we were interested to see why is it that Australia was showing Omicron variants when the country was completely locked, under a completely locked down. So, and our surprise was that we found an enormous number of haplotypes populating the makeup of Omicron, as you can see on the top. And because we had all the data, we could see what was happening in terms of haplotypes of Delta and haplotypes of Alpha. So there was a number of haplotypes, 20 in number, that we could identify. We saw that haplotypes were mutations with congruent prevalence patterns, and there you have an example, for instance, with haplotype 1 of alpha. We name it haplotype 1. But we saw a noisy rise of first haplotype in Australia, and we saw core VOC haplotypes decoupled by seasonality. So that means that genetics was somehow embedded in haplotype makeup related to seasonal behavior. And this for us was very exciting because that would imply that, that physiology is fundamental for viral makeup. So we focus on on the S protein, and we wanted to see whether there was recruitment of the haplotypes, and we saw that recruitment mirrors the 3 phases of the pandemic. That the recruitment of in the binding domain of Omicron was remarkable. It was a boom in Omicron. That haplotypes were clustered in particular regions of the S protein, meaning that there was probably crosstalk in terms of intramolecular interactions occurring, and that there were some particular clusters that were already reported by other people were related to patterns of evolution, of negative selection occurring. So therefore, there were patterns of evolution unfolding in all this that we were observing. So the conclusions of our third exploration is that variants of concern emerge, and it's an emergence, by recruitment of haplotypes and freestanding mutations as a worldwide process, not as a process of an individual place spreading the virus. We saw 3 phases of the pandemic, one linked to flexibility, rigidity, another one to environmental sensing, and another one to immune escape. This is the one that we are facing now. And then VOC haplotypes decouple with seasonal behavior. So therefore, the epidemic calendar is encoded in the viral genome. And intramolecular interactions, haplotypes are clustered in regions of the S protein matching identified in evolutionary studies, meaning that intramolecular interactions are delimiting actively through an evolutionary process, says seasonality. So the fourth exploration, and I have 10 minutes to kind of go through this, and so that we have then enough time for questions. Now we explore 12 million of these mutations in the middle of 2022. This is again a diagram. These are all diagrams from Alistair Ray in Sheffield, UK, that produces these beautiful spike diagrams. You don't need to have the continents highlighted. The population shows how population in the world lived close to the oceans. And it's quite remarkable. And again, there allows us to delimit corridors, the Arctic, northern temperate, tropic, southern temperature in the world, and use those corridors to define genomes and look for haplotypes linked to seasonality. And there you have one of them, haplotype 16 in VOC Omicron, and you can see how the haplotype appeared in the world, very consistently, and is quite distant from all the rest of that accumulation. And that was the way how we identified haplotypes. And we saw that during phase 1, there were only 2 haplotypes, isn't it? Remember the N protein link haplotype and the S protein link haplotype. But then when variants of concern appeared, then there were 20 additional haplotypes that were added to the world of mutations of the COVID-19 virus. And here from a world point of view now this is not Australia, this is the world. Again, we see Omicron harboring quite a number of those haplotypes in interaction with themselves. And as you can see, each haplotype, some of them are exclusively affecting, for instance, individual proteins like the S proteins, but others are affecting many other haplotypes. And that implies that there are protein interactions occurring between or within a protein. It could be intramolecular interactions affecting allosteric interactions, for instance, or protein-protein interactions. For instance, an S protein, the protein of the spike, interacting with the RNA polymerase protein that we saw at the beginning. So there was obviously a protein-protein interaction that was unfolding at that time. So we can see here in these diagrams, this is the diagram for alpha, delta, and omicron. The dots are the proteins, and the links imply an haplotype interaction, which is a molecular interaction. And as you can see at the beginning, everything relates to the S protein, the N protein, and this NSP6 protein, which is very interesting because the NSP6 protein is a protein involved in the secretion of pro-inflammatory cytokines in inflammasomes. And that protein is crucial because it's encoding the syndrome. Remember that syndrome that kills the patient, when the disease takes, overrides the pulmonary areas. And it's the NSP6 protein, the one that is responsible. And as you can see, it's the dot is there in the 3 networks, in alpha, delta, and omicron. But one thing that you can see here is the crucial centrality of the structural proteins, the S, the N, the membrane protein, the envelope proteins. They're all appearing and forming a tight cluster, in this diagrams. So networks link seasonal behavior to protein sequence, but do not address how mutations are linked to structure and function. This is something that we were concerned. We wanted to understand why these proteins are talking to each other. And this is where AlphaFold 2, which I will describe now, comes to play. And what is AlphaFold 2? It's called an ab initio analysis of structure. It's a software. It's artificial intelligence program for protein structural prediction that is able to generate a model of structure that is as good as the model that you would generate experimentally, through crystallography or cryo-EM, cryo-electron microscopy. These are time-consuming procedures. So people, takes months to generate one of those modules because you have to crystallize the protein and then subject it to X-ray diffraction. So if you could have a bioinformatic approach like the one I'm going to show you that produces a structure that is as good as the experimental structure, then this is wonderful because you can do it. It opens an incredible floodgate of information that you can mine. So generally what this artificial intelligence program does, it takes an input of a sequence, that's, let's say, the sequence of a spike, of a variant that you've isolated in your laboratory. And then you compare that sequence to multiple sequence alignments, like ones I showed you at the beginning of my talk, and a structure library. So instead of being only the sequences, it's also the structures that you've been aligning. So you have a library of those and a sequence of those. And these are gigantic libraries that take everything that is known. And then what you look are for covariation features that occurred, in terms of what is occurring in the alignment. And then this is fed into what is called the deep neural evoformer module, which is an artificial intelligence device that allows you to find distance and torsion distribution predictions. It predicts, what would be the likely fold of that protein. And this is then fed to another module, the structure module, that uses like an entropic measure in what is called gradient descent on protein-specific potential to really fold the structure into the real three-dimensional structure, atomic structure of the molecule. And this is the output. And now this is something new that occurred in the world of science, because for many years there's been a competition called the CASP. It's a CASP protein folding contest. And as you can see on the y-axis, you can see the average global distance test, which is a measure of how good is the alignment of something that is predicted with a reality. So you have a crystallographic entry and compare it with that is known only to you, but you provide it in the competition and see which software does well. And that has been going on for many years. And as you can see at the beginning, all the software were good for shape. But then they got good for topology of the structure. And then came AlphaFold in 2018, and which was the first version. And it produced a very good structure. And then in 22, wow, that structure was almost indistinguishable from the experimental determined structure. And this has changed the landscape of structural biology. Why? Because now with AlphaFold 2, we can study a number of things: ab initio structure prediction, construct design, protein reformation, study protein complexes, riboprotein complexes, de novo protein designs. But for me, effects of mutations in conformational space. And that's kind of the thing that we are now focusing. We want to use AlphaFold 2 to see what are all those mutations that were affecting the seasonal cycle of the virus doing at the three-dimensional level. And I'm going to show 2 slides, and I'm done with my talk. This is an illustration with the M protein. The M protein on the top there is, remember, this is the membrane protein. It has the N-terminal region with all those 3 transmembrane domains, TM1, TM2, and TM3 in pink. And then the other part is the globular part. It's inside, the protein has that globular part inside the virion capsid. It's looking inside. And this protein is fundamental. Let me tell you why it's fundamental. It's even more important than the spike protein. The spike protein is necessary to enter into the lung cells. But once the virus has, shoot the RNA inside the cell, now it has to make copies of itself. And then it has to, those copies need to now produce proteins. And the proteins will be, some of them, will be structural proteins, but the M protein is being produced. And that M protein is like a director. It directs in the membrane how the different capsids are going to be assembled with all the different components and RNA make up into that capsid. And that's why it's so important. And it turns out that the mutations that occurred in Delta and Omicron, not in Alpha, because it did not have any mutation on the M protein, were all located on the N-terminal regions. They were located in the membrane part of the molecule. But all the effects on structure, remember, all this is modeled with the AlphaFold. All the effects of structure were occurring in regions that were globular, that were looking inside the cell. So that was an interesting observation in itself. But when we explored not just the entire complement of mutations in Delta or in Omicron, but we dissected the individual haplotypes, that are making that constellation. For instance, haplotypes 14, 15, and 22 of Omicron. And we compared them. What we saw is that constellations are balancing the more extreme effect on structure of individual haplotypes. And in doing so, they're diminishing the effects that mutations have on structure. So it's like an incredible, I would say, a balance that is occurring in the structural makeup of the viruses. This was what this work is telling us, only with the M protein. So this is a non-published result that I'm going to show you now. It's only one slide, but we've done this not only for the M protein, but for all other proteins that have mutations. And we focus in particular on the spike. And all the mutations of the spike are listed here. And what we have in that diagram there on the right, we have different colors. So the more red the coloration is, the more impactful is on the structure. The mutation has been on the structure. The mutation doesn't need to be in that locale, but the mutation can affect that locale in the protein. And as you can see, there are some particular areas that are kind of important, and they are located all in the left-hand side of the sequence. And it's in the N protein, and it's in the RBD region, and then red there, like calling us. And as you can see, you have all the haplotypes below, and you have VOC delta 2 and VOC alpha. So there is a time component as the different haplotypes are appearing in the world of COVID-19 that goes from the bottom to the top. And in this analysis, you can actually explore where in the spike those mutations are appearing, and that's the three-dimensional map that you have on the left. And not only that, you can see whether they are synergistic or antagonistic patterns, occurring in terms of how haplotypes interact with each other in the constellation. And the finding for me is amazing, is that we found antagonistic patterns in alpha delta, I don't know what that is, but I don't know if you can hear that, but it must be like a clock or something. But we have antagonistic patterns invoke alpha delta, followed by synergistic patterns invoke Omicron. So that means that in Omicron, the current COVID-19 strain, mutations are talking to each other in a beneficial manner, enhancing the changes at the structural level. So what are the conclusions? A worldwide analysis confirms that the epidemic calendar is encoded in genes. Haplotypes define networks of protein interactions. They link seasonal behavior to protein sequence. And then AlphaFold, this artificial intelligence, construct, models reveal the centrality of structural proteins. They show the importance of hinged regions in the M protein, and NTD, RBD, and S1, S2 boundary regions of this protein. So all these are wonderful targets for vaccine development now. And we now find this antagonistic and synergistic patterns in structure driving evolution of haplotypes. Why is this? And this is kind of the general conclusions of the things that I've been showing. It's just repeating the take-home messages. Now, why is for me important to the synergistic and antagonistic patterns? Because I'm interested in understanding why structure in proteins have the makeup it has. We've been studying structure and how structure has been unfolding in billions of years in evolution. We've been studying how fold makeup in proteins is fundamental for determining biochemistry. What we have here with the COVID-19 virus is real-time unfolding of change, evolutionary change that we can see with our own eyes, that we don't need to go and infer with phylogenetic reconstruction approaches. Here we can basically map how time has been changing the makeup of the sequence and the structure of a protein. And we can see what are the drivers behind this. This is why we are so much interested in this kind of analysis. So, and I want to finish by showing you the people that have done the work. Trey Tomaszewski has been the one that have been involved in most of the work related to the last two of genomic exploration. Fiz and Mughal have been exploring the genomic makeup of intrinsic disorder. Kelsey, my daughter, she's in a company here in New Mexico called Call Out Biotech, has been helping a lot in bioinformatic endeavors. Asif is the one that has been doing all the AlphaFold analysis and is going to finishing his thesis soon on this. And Nicolas is an undergraduate student that did all the exploration of seasonality and links to planetary change that I showed to you that we published, and will continue to work with me on this. So thank you very much. These are the acknowledgements. Other people involved, including Volker Gertler at Melbourne, when we were running all this Australian analysis, and which was kind of very exciting when we got all those results. So thank you very much, and welcome all your questions that you may have for me. So thank you.
Thank you very much, Gustavo, for this amazing talk.